Okay, in this video we're going to be learning about different kinds of inherited disorders. So I titled this Genetic Disorders. There are a few different types, and this presentation is going to focus on two of them, the autosomal dominant and the autosomal recessive. Autosomal dominant disorders. Remember, first off, look at this name here. We have autosome. So you guys know that has to do with chromosomes 1 through 44. And these are going to be dominant disorders. So we have autosomal dominant disorders. These are disorders that occur on an autosome, and they are dominant. So that's exactly what we have right here on this definition, so jot that down. You guys know that if it's a dominant, if you have one or two of those alleles, then you're going to show the disease. So we're going to use very simple letters here. We'll use an allele like this, and then we'll use an allele like this, okay? Because this is a dominant disorder, the capital A is going to stand for the disorder. Lowercase a will stand for a normal phenotype. Okay, so that's what we're going to be focusing on in the first few slides here. Here I'm going to give you two examples of autosomal dominant disorders that affect people. You need to know the two of these as solid examples. The first one's going to be called Huntington disease. Every human makes a protein called Huntington. What this Huntington protein is supposed to do is it helps brain cells to develop properly and to function properly. So because this is a autosomal dominant disorder, if someone, we'll say capital H stands for the disease phenotype, and lowercase h stands for normal. So if someone gets a single mutated copy of this gene, so even if they're heterozygous, they get Huntington's disease. This disorder, as far as the phenotype here, there's really no treatment. People don't know they have Huntington's disease until they're middle age, probably into their 40s, maybe late 30s. It causes the brain cells to degenerate, nerves to degenerate. People's brains can't communicate with their body. It seems like they have personality disorders because their brain isn't functioning properly. They lose control of their muscles in their body because the nerves are breaking down. And what's actually causing it is because these abnormal Huntington proteins are being made by that defective DNA, they're clumping together inside the nerve cells, and the nerves tolerate that to a point. But at a certain point, they do pile up, and they cause the nerves not to work properly. Because there are no symptoms seen until someone reaches middle age, a lot of people have already had kids by then, and they have already risked passing this on to children. So this is absolutely something that runs in families. If this ran, for instance, in your family, you would absolutely know about it. And we'll see why in just a minute. So the phenotypes that would be, genotypes that would be affected, if someone got an allele from each parent, or if they got an allele from one parent, these would show the disease. In families that have Huntington disease, this is the only way to not have the disorder, to receive recessive alleles from each parent. Another example of an autosomal dominant disorder that I want you to know is called achondroplasia. If you've ever seen the show Little People, Big World, this was the particular disorder they had. This is one of the many ways to suffer from dwarfism. And what this is, it's a dominant. So at least one defective gene is inherited that lets the long bones grow to the regular length. Long bones would be like your humerus in your upper arm, your radius and ulna in your forearm, your femur in your upper leg, and your tibia and fibula in your lower leg. Those are your long bones. They don't grow to proper length, and that's why we see this. Torso and head, they're not made of long bones, so they grow to normal size. What we have found with this is if a baby is conceived and got a dominant allele from both parents, that baby doesn't survive long enough to be born. That would be a miscarriage. The people that are heterozygous are the ones that suffer from this disorder. And in a family that this was running in, if a child had small a, they were homozygous recessive, they would be of normal height in that family. Okay, let's try this out here. Let's cross a man with Huntington's disease with a normal woman. And let's just say the man is, let's say, heterozygous for his Huntington's disease. With a woman, it does not run in her family. And we want to know what the probabilities of having children with the disease are. Well, this is how we do that. So the man is going to be heterozygous for his Huntington's disease. And the woman does not have Huntington's disease. So this will be her genotype. We do the Punnett square like always, but now we're not predicting for a certain trait like hair color. Whoops. What we're doing is we're looking at inheriting a disease or not. Punnett square looks just like anything else, but it's just going to show us something a little bit different. So we know as far as genotypes go, we're seeing this genotype in 2 out of the 4 probability here. And over here, that would be another 2 out of 4. And then what does this actually mean? These individuals here, they have the disease. Or that's the chance of having kids with the disease. So again, that's still 2 out of 4. And down here, this would be not having the disease. So the chance of that is 2 out of 4 as well. So if these two people do have children, and whether the man knows he has the disease or not, they have a 50-50 chance of having kids affected with this Huntington's disease or not being affected with this Huntington's disease. It would be the same kind of Punnett square for achondroplasia, where the dominant is causing the problem. So remember that one allele, that will be for our disease phenotype, and this will be for normal. And because this is a dominant disease, getting 1 or 2 of these is going to lead to having the disease. Okay, here's a different type of genetically inherited disease of the autosomes. So this is a gene found somewhere on chromosomes 1 through 44, depending on which one we're talking about. We're not talking about those sex chromosomes. And this is a recessive disorder. Okay, so that's what we have here. You all know because of this, if we let this, it's going to be reversed now. This is going to stand for normal DNA because this is a recessive disorder, a problem with a recessive allele. So here, if a person is like this, they do not have the disease that runs in their family, if it's a recessive disease. If someone's heterozygous, we're going to give them a special designation. They're normal, but we're going to call them a carrier. That means they have this defective piece of DNA right here. It's not making them have the disorder, but they could potentially pass that on to a child if they were to, they could have a child with the disease if they were to marry someone else who that same disease ran in their family. And in this case, this would be a disease phenotype. Okay, so make sure you write all that down, please. Here's 2 that I want you to know about. Tay-Sachs disease and cystic fibrosis. Tay-Sachs disease, people need to make a protein called hexa that breaks down certain types of fat in cells. People that have Tay-Sachs disease, they don't live very long. They have brain problems, motor problems, they can't move their body quite as well. We see this by 8 months. Those babies aren't hitting these normal developmental milestones they're supposed to hit. And by 3 to 4 years old, they can't tolerate this anymore. You guys know this from studying cells, that because they can't break down the fat, it builds up in the lysosomes and impairs their function. Because the lysosomes then can't break down materials, because it's unable to break down these fats, it's now, the nerve cells are especially susceptible to that. And like it says here, the brain cells are affected the most. So again, in families, this would be a normal genotype. This would be the carrier. And these here would be the individuals that have the disease. Another disorder is called cystic fibrosis. You may have heard of this one. Every person needs to make a transmembrane protein that transports chloride, so this charged ion, across the plasma membrane. People with cystic fibrosis, remember, they don't make any of that proper chloride channel. This affects the lungs and the pancreas the most. The pancreas produces enzymes to help you break down your food. So because the pancreas can't function properly, these people have a hard time getting enough calories out of their food because they're not breaking it down all the way, have a really hard time maintaining body weight. The lungs are very susceptible, and because this chloride can't be moved around, in and out of cells, thick mucus builds up in the lungs. It can become infected, can limit breathing, can cause chronic lung infections. This age here may be a little outdated. I think with more medical advances, this is probably more so in the 40s by now. Another example of an autosomal recessive disorder, you don't really need to know, just know these two would be albinism. Okay, it's a disorder where the body doesn't produce any pigment in the hair, skin, or eyes. And I know that you've probably heard of that one. So that would be a normal, non-albino individual. This would be a person who has it in their family, but they're just a carrier. And lowercase, that homozygous recessive would be the people that have albinism. So, let's do, well, first off, here's the definition of what a carrier is in these autosomal recessives. So please write down this definition. An individual who has one allele for an autosomal recessive disorder as well as one normally functioning allele. They are not showing the phenotype for the disorder because the one normally functioning, that dominant, is enough to make the phenotype normal. And this is a very interesting video on cystic fibrosis here. We'll watch that in class. Here's a practice problem. A carrier for cystic fibrosis and a normal non-carrier want to have children. What are the possible phenotypes of their children with regard to cystic fibrosis? So let me get to a blank screen. Here's our carrier. And here's someone who is not a carrier. They do not have the disorder either. So we'll say normal, non-carrier. This is the reverse of the autosomal dominance as far as what capitals and lowercases stand for, so you have to identify these properly. Put our carrier here, our non-affected individual on the side. As far as genotypes go, we're seeing a 50-50 here. And then we have to interpret this now. We have to know what these mean. Phenotypes, these would be normal non-carriers. Say, not afflicted. 2 out of 4. And these here, they are not afflicted either, but they're carriers. Okay, and that's how we'll set that one up to do it. Okay, so to review, let me come back to this slide here. We went through 2 different types of inherited disorders. We talked about some that are found on genes found on autosomal chromosomes, chromosomes 1 through 44. Autosomal dominant is a defective dominant allele, and we went through what that was and some examples, and then we did a Punnett square of one to predict offspring. The other example we did was autosomal recessive. That is where the recessive alleles are the cause of the problem, and we can have carriers in that situation. So those were the 2 examples that we had, and we'll be doing some practice problems with these during our next class.